This is the last video in which we walk through an example calculating allele frequencies given genotypic frequencies and calculating expected genotypic frequencies given the allele frequencies. This video goes over our takeaways. Let's say we have a population. Within that population, we can look at some different levels. We can look at the phenotype, shown on the right, since Indy's phenotype is that she does have hind dew claws. We can also look at the genotype for that trait. In Indy's case, her genotype is CT. Or we can look at an even smaller scale, zoomed in, if you will, and look at the alleles that we see. And we can look at all of these within the greater framework of this population. For example, at what frequencies do we see each phenotype in the population? What are the genotype frequencies in the population? And even what are the allele frequencies in the population? If we know what the genotype frequencies of the population are, we can use those to calculate the allele frequencies. In this case, we use the allele counting method, as we demonstrated in video 2, but there are also other methods available. If we don't know the genotypes, we can use the phenotypes to estimate the allele frequencies in the population. For example, if we know how many dogs do have hind dew claws, then we know the number of dogs that are CT or TT. However, we don't know which ones are homozygous and which ones are heterozygous. The dogs without a hind dew claw are CC, or homozygous recessive. We can solve this using the square root method, but it's less precise. Once we have the allele frequencies, we can use these to calculate what we expect the genotype frequencies to be if the population is in Hardy-Weinberg equilibrium. And then as we talk through some of these examples, we can also think about the fact that phenotype is actually not just a result of the alleles we see at this locus or for this gene. In fact, each phenotype or an individual's entire phenotype is usually going to be determined by lots and lots of genes as well as the environment. And some genes are more impacted by environment than others. Likewise, some may be primarily controlled by genes. The point here is that if you know one or two of these things, you can likely figure out the others. And you can use these to determine whether a population is in Hardy-Weinberg equilibrium or whether it is evolving. If you're curious about this, here are some scientific articles. Specifically, these are about dew claws in dogs. Also, I have a link to Embark here at the bottom, and if you're interested in getting your dog's DNA tested, there's a discount code there. Of course, if you have any questions, go ahead and join us for office hours or post to the discussion boards. Thanks for watching.